Class 11, Chapter 13, Plant Growth and Development. You have already studied the organization of a flowering plant in Chapter 5. Have you ever thought about where and how structures like roots, stems, leaves, flowers, fruits, and seeds arise, and that too in an orderly sequence? You are, by now, aware of the terms seed, seedling, plantlet, mature plants. You have also seen that trees constitute to increase in height or girth over a period of time. However, the leaves, flowers, and fruits of the same tree not only have limited dimensions but also appear and fall periodically and sometimes repeatedly. Why does vegetative phase precede flowering in plant? All plants' organs are made up of a variety of tissues. Is there any relationship between the structure of a cell, a tissue, an organ, and the function they perform? Can the structures and the function of these be altered? All cells of a plant are descendants of the zygote. The question is then, why and how do they have different structural and functional attributes? Development is the sum of two processes, growth and differentiation. To begin with, it is essential and sufficient to know that the development of a mature plant from a zygote, fertilized egg, follow a precise and highly ordered succession of events. During this process, a complex body organization is formed that produce roots, leaves, branches, and flowers, fruits, and seeds, and eventually they die. The first step in the process of plant growth is seed germination. The seed germinates when favorable conditions for growth exist in the environment. In absence of such favorable condition, the seeds do not germinate and goes into a period of suspended growth or rest. Once favorable conditions return, the seeds resume metabolic activities and growth takes place. In this chapter, you shall also study some of the factors which governs and controls the developmental process. These factors are both intrinsic and extrinsic to the plant. Now growth. Growth is regarded as one of the most fundamental and conspicuous characteristic of a living being. What is growth? Growth can be defined as an irreversible permanent increase in size of an organ or its part or even of an individual cell. Generally, growth is accompanied by a metabolic process, both anabolic and catabolic, that occur at the expense of energy. Therefore, for example, expansion of a leaf is growth. How would you describe the swelling of a piece of wood when placed in water? Now, plant growth generally is indeterminate. Plant growth is unique because plant retains the capacity for unlimited growth throughout their life. This ability of the plant is due to the presence of meristems at certain locations in their body. The cells of such meristem have the capacity to divide and perpetuate. The product, however, soon loses the capacity to divide, and such cells make up the plant body. This form of growth, wherein new cells are always being added to the plant body by the activity of meristem, is called open form of growth. What would happen if the meristem ceased to divide? Does this ever happen? In earlier classes, you have studied about the root apical meristem and the shoot apical meristem. You know that they are responsible for the primary growth of the plants and principally contribute to the elongation of the plants along their axis. You also know that in dicotyledonous plant and gymnosperms, the lateral meristem, vascular cambium, and cork cambium appears later in life. These are the meristems that cause the increase in girth of the organs in which they are active. This is known as secondary growth of the plant. Growth is measurable. Growth at cellular level is principally a consequence of increase in the amount of protoplasm. Since increase in protoplasm is difficult to measure directly, one generally measures some quantity which is more or less proportional to it. Growth is therefore measured by a variety of parameters, some of which are increase in fresh water, dry weight, length, area, volume, and cell number. You may find it amazing to know that one single maize root apical meristem can give rise to more than 17,500 new cells per hour, whereas cells in a watermelon may increase in size by up to 350,000 times. In the former, growth is expressed in increase in cell number. The latter expresses growth is increase in size of the cell. While the growth of a pollen tube is measured in terms of length and increase in surface area denotes the growth in a dorsiventral leaf. Phases of growth. The period of growth is generally divided into three phases, namely meristematic, elongation, and maturation. Let us understand this by looking at the root tips. The constantly dividing cell both at the root apex and the shoot apex represent the meristematic phase of growth. The cells in this region are rich in protoplasm, possess large conspicuous nuclei. Their cell walls are primarily in nature thin and cellulosic with abundant plasmodesmatal connection. The cells proximal to the meristematic zone represent the phase of elongation. Increased vacuolation, cells enlargement, and new cell walls deposition are the characteristic of the cells in this phase. Further away from the apex, that is more proximal to the phase of elongation, lies the portion of axis which is undergoing the phase of maturation. The cells of this zone attain their maximal size in terms of wall thickening and protoplasmic modification. Most of the tissues and cell types you have studied in your earlier classes represent this phase. Growth rate. The increased growth per unit time is termed as growth rate. Thus, rate of the growth can be expressed mathematically. An organism or a part of the organism can produce more cells in variety of ways, arithmetic and geometric. Now, the growth rate shows an increase that may have arithmetic or geometric. The arithmetic growth following mitotic cell division, only one daughter cell continue to divide while the other differentiates and matures. The simplest expression for arithmetic growth is exemplified by root elongation at constant rate. Look at the figure 13.5 here given. On plotting the length of organ against time and linear curve is obtained. Mathematically expressed term given below. Let us now see what happens in geometrical growth. In most systems, initial growth is slow, the lag phase, and it increases rapidly thereafter at an exponential rate. Here, both the progeny cells following mitotic cell division retain the ability to divide and continue to do so. However, with limited nutrient supply, the growth slows down, leading to a stationary phase. If we plot the parameter of a growth against time, we get a typical sigmoid or S curve. A sigmoid curve is a characteristic of living organism growing in a natural environment. It is typical for all cells, tissues, and organs of a plant. Can you think of some more similar examples? What kind of a curve can you expect in a tree showing seasonal activities? The exponential growth can be expressed in this way. So here R is the relative growth rate and also the measure of the ability of the plant to produce new plant material, referred to as efficiency index. Hence, the final size of W1 depends on the initial size W0. Quantitative comparison between the growth of the living system can also be made in two ways. First, measurement and comparison of total growth per unit time is called absolute growth rate, and the second, the growth of the system given system per unit time expressed on a common basis. Example, per unit initial parameters is called relative growth rate. In figure 13.7, which is given above, two leaves A and B are drawn that are of different size but shows absolute growth rate in areas in the given times to give leaves A1, B and B1. However, one of them shows much higher relative growth rates. Which one and why? You have to find it out. Now, condition for the growth. Why do you not try to write down what you think are a necessary condition for growth? This list may have water, oxygen and nutrients as well as essential elements for growth. The plant cell grow in size by cell enlargement, which in terms requires water. Turgidity of cells helps in extension growth. Thus, plant growth and further development is intimately linked to the water status of the plant. Water is also provide and medium for enzymatic activities needed for the growth. Oxygen helps in releasing metabolic energy essential for growth activities. Nutrients, macro and micro essential elements are required by plants for the synthesis of protoplasm and act as source of energy. In addition, every plant organism has an optimum temperature range best studied for its growth. Any deviation from this range could be detrimental to its survival. Environmental signals such as light and gravity also affect certain stage of growth. Now, differentiation, dedifferentiation and redifferentiation. The cells derived from root apical and shoot apical meristem and cambium differentiate and mature to perform a specific function. This act leading to the maturation is termed as differentiation. During differentiation, cells undergo few to major structural changes both in their cell walls and protoplasm. For example, to form a tertiary element, the cells would lose their protoplasm. They also developed a very strong elastic lignocellulosic secondary cell walls to carry water to long distance even under extreme conditions. Try to correlate the various anatomical features you encounter in plants to the functions they perform. Plants show another interesting phenomenon. The living differentiate cells that by now have lost the capacity to divide can regain the capacity of cell division under certain conditions. This phenomenon is termed as de-differentiation. For example, formation of meristems, interfascicular cambium, cork cambium from fully differentiated parenchyma cells. While doing so, such meristem tissues are able to divide and produce cells that once again lose the capacity to divide but matures to perform a specific function, that is differentiation. List some of the tissue in a woody dicotyledonous plant that are the product of redifferentiation. How would you describe a tumor? What would you call the parenchyma cells that are made up to divide under controlled laboratory condition during plant tissue culture? Recall in section 13.111 we have mentioned that the growth in plants is open, that is, it can be indeterminate or determinate. Now we may say that even if differentiation in plants is open, because cells tissue arising out of same meristem have different structures at maturity. The final structures at maturity of a cell tissue is also determined by the location of the cells within. For example, cells positioned away from the root apical meristem differentiate as root cap cells, while those pushed to the periphery mature as epidermis. Can you add few more examples of the open differentiation correlating the position of the cell to its position in an organ? Development. Development is term that includes all changes that an organism doesn't goes through during its life cycle, from germination to seed to senescence. Diagrammatic representation of the sequence of the process which constitute the development of a cell of a higher plant is given in figure 13.8. You can see it is also applicable to tissue or organ. These are the figures which are given here, 13.8. Plant follow different pathways in response to environment or phase of the life to form different kind of structures. This ability is called plasticity. Heterophyly in cotton, coriander and larkspur. In such plants, the leaves of the juvenile plants are different in shape from those in mature plants. On the other hand, difference in shapes of the leaves produced in air and those produced in water in buttercup also represent the heterophilous development during environment. This phenomenon of heterophyly is an example of plasticity. Thus, growth, differentiation and development are very closely related events in their life of the plants. Broadly, development is considered as the sum of the growth of and differentiation. Development in plant, that is both growth and differentiation, is under the control of intrinsic and extrinsic factor. The former includes both the intracellular or intercellular factors such as plants growth regulators, while the latter includes light, temperature, water, oxygen and nutrients, etc. Now, the plant growth regulator characteristic first we will discuss. The plant growth regulator are small simple molecules of diverse chemical composition. They could be indole compounds, adenine derivatives and derivatives of carotenoids, terpenes or gas such as ethylene C2H4. Plant growth regulator are variously described as plant's growth substance, plant hormones or phytohormones in literature. The PGR plant growth regulators can be broadly divided into two groups based on their functions in a living plant body. One group of plant PGRs are involved in growth promoting activities such as cell division, cell enlargement, pattern formation, tropic growth, flowering, fruiting and seed formation. These are also called the plant growth promoters. Example, auxins, gibberellins and cytokinins. The PGRs of the other group play an important role in plant response to wound and stress of biotic and abiotic origin. They are also involved in various growth inhibiting activities such as dormancy and abscission. The PGR abscisic acid belongs to this group. The gaseous PGR ethylene could fit either of the group, but it is largely in an inhibitor or growth activities. The discovery of plant growth regulators. Interestingly, the discovery of each of the five major groups PGR have accidental. All these started with the observation Charles Darwin and his son Francis Darwin when they observed that coleoptiles of canary grasses respond to unilateral illumination by growing towards the light source photoperiodism. After a series of experiments, it was concluded that the tip of coleoptile was the site of transmittable influence that caused the bending of the entire coleoptile. Auxin was isolated by F.W. Wendt from tips of coleoptiles of oat seedlings. The Bakane fooling seedling disease of rice seedling was caused by a fungal pathogen Gibrella fujikuri. E. Kurosawa 1926 reported that the appearance of the symptoms of the disease in rice seedling when they were treated with the sterile filtrates of the fungus, the active substance were later identified as gibberellic acid. F. Scu and his co-workers observed that from the internodal segments of tobacco stems and callus, a mass undifferentiated cells proliferate only if an addition to oxygen and nutrients medium was supplemented with one of the following extracts of vascular cambium, yeast extract, coconut milk, or DNA. Miller et al. 1955 later identified and crystallized the cytokinesis promoting active substance that they termed kinetin. During mid 1960s, three independent researchers reported that the purification and chemical characterization of three different kinds of inhibitors, inhibitor B, abscission 2, and dormin. Later, all the three were proved to be chemically identical. It was named abscisic acid, ABA. H. Cousin, 1910, confirmed that the release of the volatile substance from the ripened organs that hastened the ripening of the stored unripened bananas. Later, this volatile substance was identified as ethylene and a gaseous PGR. Let us study some of the physiological effects of these five categories of PGR in the next section. First, auxin. Auxin, from Greek word auxin, means to grow, was first isolated from human urine. The term auxin is applied to the indole 3 acetic acid and to other natural and synthetic compounds having certain growth regulating properties. They are generally produced by the growing apices of the stems and roots, from where they migrate to the regions of their action. Auxins like IAA and indole butyric IBA acid have been isolated from plants. NAA and 2,4-D dichlorophenoxyl acetic are synthetic auxins. All these auxins have been used extensively in agriculture and horticulture practices. They help to initiate rooting in stem cutting and application widely used for plant propagation. Auxins promote flowering in pineapples. They help to prevent fruiting and leaf drop at early stage but promote the abscission of older mature leaves and fruits. In most higher plants, the growing apical bud inhibits the growth of the lateral axillary bud, a phenomenon called apical dominance. Removal of shoot tips decapitation usually results in the growth of the lateral buds. It is widely applied in tea plantation, haze making. Can you explain why? Auxins also induce parthenocarpy, that is in tomatoes. They are widely used as herbicides. 2,4-D widely used to kill dicotyledonous weeds, does not affect mature monocotyledonous plants. It is used to prepare weed-free lawns by gardeners. Auxin also controls xylem differentiation and helps in cell division. Now second, gibberellins. Gibberellins, another kind of promotory PGR. There are more than 100 gibberellins reported from widely differentiated organisms such as fungi and higher plants. They are denoted by GA1, GA2, GA3 and so on. However, gibberellic acid was one of the first gibberellins to be discovered. GA3 was the first gibberellins to be discovered and remains the most intensively studied form. All gibberellic acid are acidic. They produce a wide range of physiological response in the plants. Their ability to cause an increase in length of axis is used to increase the length of the grapes stalks. Gibberellins cause fruits like apples to elongate and improve its shape. They also delay senescence. Thus, the fruit can be left on the trees longer so as to extend the market period. GA3 is used to speed up the melting process in brewing industry. Sugarcane stores carbohydrate as sugars in their stems. Spraying sugarcane crops with gibberellins increase the length of the stem, thus increase the yield as much as 20 tons per acre. Spraying juvenile conifers with gibberellic acid hasten the maturity period, thus the leading to early seed production. Gibberellins also promotes bolting, internode elongation just prior to flowering in beet, cabbage, and many plants with rosette habit. Cytokinin. Cytokinin have a specific effect on cytokinesis and were discovered as kinetin, modified form of adenine and purine from autoclaved herring sperm DNA. Kinetin does not occur naturally in plants. Such search for natural substances with cytokinin-like activities led to the isolation of gelatin from corn kernels and coconut milk. Since the discovery of gelatin, several naturally occurring cytokinesis, kinin, and some synthetic compounds with cell division promoting activity have been identified. Natural cytokinins are synthesized in regions where rapid cell division occurs. For example, root apices developing shoot buds, young fruits, etc. It helps it to produce new leaves, chloroplast in leaves. Lateral shoots growth and adventitious shoots formation. Cytokinin help overcome the apical dominance. They promote nutrients metabolism which help in the delay of the leaf senescence. Now ethylene. Ethylene is a simple gaseous PGR. It is synthesized in large amounts by tissue undergoing senescence or ripening fruits. Influence of ethylene on plants include horizontal growth of the seedling, swelling of the axis, and apical hook formation in dicot seedling. Ethylene promotes senescence and abscission of the plant's organs, especially of the leaves and the flowers. Ethylene is highly effective in fruit ripening. It enhances the respiration rate during ripening of the fruits. This rise in rate of the respiration is called respiratory climactic. Ethylene breaks seed and bud dormancy, initiates germination in peanut seeds, sprouting of potato tuber. Ethylene promotes rapid internode petiole elongation in deep water rice plants. It helps leaves upper parts of the shoot to remain above water. Ethylene also promotes root growth and root hair formation, thus helping the plants to increase their absorption surface. Ethylene is used to initiate flowering and for synchronizing fruit set in pineapples. It also induces flowering in mango. Since ethylene regulates so many physiological processes, it is one of the most widely used PGR in agriculture. The most widely used compound as source of ethylene is ethifone. Ethifone in an aqueous solution is readily absorbed and transported within the plant and releases ethylene slowly. Ethifone hastens fruit ripening in tomatoes and apples and accelerates abscission in flowers and fruits, thinning of the cotton, cherry, walnut. It promotes female flowers in cucumbers, thereby increasing the yield. Now, the fifth one, abscisic acid. As mentioned earlier, abscisic acid ABA was discovered for its role in regulating abscission and dormancy. But like the other PGR, it also has the wide ranging of effect on plants' growth and development. It acts as a general plant inhibitor and an inhibitor of the plant metabolism, inhibits the seed germination, stimulates the closure of stomata, and increases the tolerance of plants to various kinds of stress. Therefore, it is also called the stress hormone. Abscisic acid plays an important role in seed development, maturation, and dormancy by inducing the dormancy. Abscisic acid helps seed to withstand desiccation and other factors unfavorable for the growth. In most situations, abscisic acid acts as an antagonistic to gibberellic acid. We may summarize that for any and every phase of growth, differentiation, and development of plant, one of the other PGR has some role to play. Such roles could be complementary or antagonistic. These could be individualistic or synergetic. Similarly, there are a number of events in the life of a plant where more than one PGR interact to affect the events. Is a dormancy in seeds, buds, abscission, senescence, apical dominance, etc. Remember, the role of PGR is only one kind of intrinsic control along with the genomic control and extrinsic factors. They play an important role in plant growth and development. Many of the extrinsic factors such as temperature and light control plant growth and development via PGR. Some of such events could be vernalization, flowering, dormancy, seed germination, and plants movements, etc. We shall discuss briefly in the role of the light and temperatures. Both of them are extrinsic factors on initiating the flowers. So that's it. Thank you so much.